Okay, hi everybody. Dr. Fasciano here with my assistant Grace, and we are going to be doing a brain dissection. So I'm going to post this video as a demonstration for you, and then when we meet on Wednesday, you guys will be doing some of this. So the package came with a sheet, a poster like this that's got a lot of the structures. And what I plan to do in this demonstration is go through this sheet with you guys and point out a lot of these structures as we are dissecting the brain. So you can see that you have a little dissection kit that came in your box. So you should have the forceps, the tweezers, you have scissors, you have a scalpel, and you have some dissection pins, and you have a probe, which is cool. The probe is good for pointing at things. And so as I'm going through this brain, I'm going to be kind of holding it up at different times and maybe even angling the camera down as I'm trying to figure out exactly the best way to get good shots for you guys. So I guess we can go ahead and we can cut open this bag. Now, as you can see, I'm in my basement at a work table. I've got the sheet laid out. It's kind of like a puppy pad, if you guys have ever puppy trained anybody. Kind of like a puppy pad, so that'll get any liquid that's in here. You'll notice there's not a lot of liquid in here, but there's a little bit. So what you want to do is kind of cut the top and keep the thing upright a little bit so that it doesn't really spill all over the place. If it does, it's no big deal. It's not going to harm you or anything like that. I will be posting a sheet along with this dissection that talks about what the chemical is that they use. They do preserve it in formalin, but then they rinse it with some sort of proprietary thing that gets rid of almost all the formalin. So there's really not anything that's cancerous in there. So I'm going to cut this open with the scissors. Okay, and you're going to take the brain out. So this brain has the dura mater on it, and I know we haven't had a chance to do our lecture on the brain yet, but there are 3 layers of protective membranes that surround the brain and protect it, and those layers are called meninges, right? When people get infections in those layers, they get meningitis. And the outermost layer is the toughest. It is called the dura mater. And dura is from the same root as durable, and so it's tough. It's hard to see here, but it's this glassy layer. It almost looks like leather when you feel it. It doesn't feel that tough until you start to pull this off. So this little thing is a sheep's brain, right? And it's got the dura mater on it. It's got that tough layer. So that thing is kind of clear and see-through, and you can see that it covers the entire brain. And what we need to do is remove that in order to see a lot of these structures that are on the listing. Now, the way to remove it is to take your scissors and kind of cut away at it. And what you'll notice, and again, it's kind of hard to see with the lighting down here, what you'll notice on the underside of the brain, there are these two big areas, these 2 big things. Those are olfactory bulbs. Okay, so those are connected to your first cranial nerve. Those are for your sense of smell. So you don't want to rip those off. You have to be careful when you're getting around the bottom here. This is where let's see right here. This is where your optic nerves come in to your brain. So you don't want to rip those off. So it's easier to start kind of at the top. What you can do is kind of poke through here with your scalpel, right? And even I'm just doing it with a probe. You can poke through there. It's kind of a skinny, a thin layer, a thin clear layer. And then what you can do is get your scissors underneath that and start cutting. Okay, now what you don't want to do is pull the bottom off, because you'll pull a lot of these structures off of the bottom. You want to try to preserve that as best you can. So my assistant here is going to cut away some of that. And once you get a good cut going, you can actually, get your fingers in there and pull a lot of that off the top. Now, again, you just want to be careful when you're pulling it around the side. So pull it towards the front of the brain, towards the anterior side. Nope, the other side. There you go. Good. All right, so my assistant started, so you can pull, you can be a little bit rough with this. Just try not to be too rough when you go around to the bottom of it. Okay, so you can see the brain is coming out nice. You can see a lot of the structures in there. Now, when you get to the point where you are nearing the cerebellum, you just have to be a little bit careful and cut it. And see now, this dura mater came off. It's pretty thick. I know it doesn't seem real thick. It seems, you know, kind of thin, but it's pretty thick and pretty hardy to get through. What we're left with is the brain, right? This is still has the two other meninges on there, the arachnoid mater and the pia mater. The arachnoid mater, arachna reminds you of a spider. The arachnoid mater gets down into all the little crevices. So it looks like a spider web. It's all the dark lines that go down into the crevices. And underneath that, the really thin layer is called the pia mater. And what's trapped between the arachnoid mater and the pia mater is cerebrospinal fluid. It's fluid, and that's what gives it these black lines. It's actually fluid that once it gets preserved, it turns into these black lines, which are kind of hard to see here. But what I want you to do is take your sheet that came with this. There's a nice blue card that came with this. And there's one side of it that's labeled 1 through 20. And it has a bunch of structures labeled that are looking at the whole brain, okay? And there's not anything that's cut at this point, right? You want to look at these structures of the whole brain. So this big structure, again, it's kind of hard to see. This whole big structure is the cerebrum, right? That's where a lot of our higher brain thoughts come from. This little structure on the back is called the cerebellum. So I'm just going in order here. Number 3 is the spinal cord coming off the back of the brain. 4 is the frontal lobe, so 4, 5, 6, and 7 are lobes of this cerebrum, right? So the cerebrum is such a large part of the brain that they actually divide it into lobes. Instead of just calling it one cerebrum, there's actually lobes of it. And the lobes are named for the skull bone. So if you remember the skull bones that go over this, you'll know the names of the lobes. So the frontal lobe is at the front. That's where your forehead would be. Right? Behind that is your parietal lobe. In the back of the brain is the occipital lobe. And then on the side of the brain is the temporal lobe. Okay? You'll also notice that the brain, if you look at it from the top, the cerebrum is divided in half, right? So you have a left hemisphere and a right hemisphere. Left hemisphere, right hemisphere. And you have a line going right down the middle. That line down the middle is the medial longitudinal fissure. So a fissure is just a crack, a crevice down the middle. That's where the brain stem. So the brain stem, if we look at the brain from the bottom now, so now I'm looking at this from the bottom, you can see that kind of the continuation of the spinal cord. So the spinal cord was here. Continuation of that is the brain stem. The medulla is this long part, the medulla oblongata. The pons is kind of this rounded part that sticks out, this bulge right here. The optic tract is cut off. The optic chiasma are these two little stumps that go into the brain, okay, and they cross. It looks like a little X because if you guys know anything or have heard anything about your eyes, it is true, whatever you see with your right eye goes to the left side of your brain. Whatever you see with your left eye goes to the right side of your brain. So that's right where those two optic nerves cross over each other. The olfactory bulbs are near the anterior part of the brain. Right? I can get my pointer under one of them here. Olfactory bulb 1, here's the other olfactory bulb on the other side. Those are obviously for your sense of smell. Those are related to cranial nerve number 1. Your midbrain is really just the region kind of at the top of the medulla oblongata. So here's your medulla oblongata. Your midbrain is right there at the top. And then we have sulci and gyri. So sulci and gyri are just fancy words for the valleys and the peaks. On the other side of the sheet, you have a nice little glossary. And it tells you that the gyri are the bulges in the fold, and the sulci are the furrows. And so what that means is that you have all these little crevices in your brain, right? All these wrinkles. The mountains, the peaks, are the sulci, right? Mm-hmm. Yeah. No, they're the gyri. The bulges are the gyri. I always get that mixed up. So the gyri, the bulges, the mountains are the gyri. The valleys, the little bridges in between them, are the sulci. And you'll, as we go through the chapter, there are certain ones that are named that are a little more prominent than others in the human brain. So that's all that those are referring to: the bulges, or the peaks, and the valleys. So then, if we flip this picture over, there is a picture of this brain cut in half, and there's a lot of structures in there. And so what you're going to do is you're going to kind of start with this cerebral hemisphere, and you're just going to kind of separate the two with your hands a little bit. Maybe even take the probe and separate them, because they are going to be separated at the top pretty well. Okay. You can even take your hand and just kind of separate it a little bit like that before you cut into it. Now realize that that means the left and right cerebrum are only attached way down there. Okay, they're not attached up here at the top. Not attached to each other, I should say. Can you hit pause on? We're just going to pause for 1 second. We'll be right back. Yeah, I have to pause. So, and then share the camera. Yep, there you go. All right, we're back. So we were looking at all the external structures, which we did. We kind of pulled apart these two cerebral hemispheres. So you can see now very clearly what that fissure is. It's that crack between them. The other thing I want to do is you pull down. So here's the brain. We're going to look from the back. And you're going to pull down kind of where the cerebellum meets the cerebrum. And when you look in there, there are 4 little lumps. 1, 2, 3, and 4. Those are called the superior, there's 2 superior colliculi right there, right there. And then you have 2 inferior colliculi down below it. Together, that's called the corpus quadrigemina. And that's something that's on the handout that I'll send around to you guys, that I'll send to you when I'm posting this video. It doesn't really say that on here anywhere, but that's what's on the handout that kind of says, oh, cut here and look for this and look for that. So what you want to do is take your scalpel, and you're going to want to cut all the way down the middle of this. So all the way down the middle of this, and even through the middle of the back here, which is the cerebellum, and then the pons, pons and the medulla. So I'll have my assistant do that.
You there?
Keep going, yep. Perfect. Now, you will have, I'll take this half, I guess. This half looks just like the picture in your book, okay? You can see a lot of the structures in there. A lot of them we've already named. You can see the cerebrum. You can see how big the cerebrum is now. And what you'll see is this one tract of tissue, which is, I don't know, the lighting is so bad in here. It might help a little bit. Yeah, that's good. Thank you. There is this one track in here that's the only place where the right cerebral hemisphere meets the left cerebral hemisphere, and that's called the corpus callosum. So that thing that you cut through is the only place where any messages from the right cerebral hemisphere get to the left cerebral hemisphere. So you got to make sure that you can find that. You can find the corpus callosum, which is number 31 on this diagram. The other things that you want to see in here, if we look at the, this is good right here. If you look at the, if you look at the cerebellum, okay, if you look at the cerebellum, there's what looks like a tree in there. It looks like a white tree in there. And that's called the arbor vitae. That's the white matter in the brain. And arbor vitae literally means tree of life. So it looks like the tree of life in there. You can see it much better. It's hard to see on the actual demonstration, but you'll be able to see it on yours much better. So you can see clearly you will be able to see the spinal cord, the medulla oblongata, the rounded pons. This is the cerebellum on the back with the arbor vitae. You'll be able to see the cerebrum. You'll be able to see the corpus callosum. I'm just going through my page real quick to make sure I can see everything. The ventricles we'll talk about. You can't really see the ventricles on a dissection. The ventricles are where you make cerebral spinal fluid. And right in between the corpus callosum, there's a kind of a little space where there's some brown or some black, whatever you want to call it. And that is the third ventricle, okay? And the black or the brown stuff is actually the cerebral spinal fluid that's in there. The formaldehyde, or it's not formaldehyde, formalin, actually preservative, turns it that color, which is also why you have that brown or black color in all these folds. That's cerebral spinal fluid in there. So I'm going to actually leave it at this for now, okay, since seeing how bad the lighting is in here. Or I don't think it's actually just in here. I think it's the general lighting on the computer. But I'm going to leave this demonstration like this for now. And then what we'll do is we'll meet on Wednesday, and I'll have you guys do some dissections in front of me. So be able to set up on Wednesday wherever you can do some dissections, and then we can go through some of these together. Okay, I'm going to save this specimen, maybe. I won't tell my wife. I'll save this specimen in a Ziploc bag for a couple of days so that I can use this to kind of show you guys what some of the things are on Wednesday as well. Okay, so I will post this for you tonight along with an email, and I will see you guys all on Wednesday.